other disciplines. He also works remotely as an adjunct professor of biology at... All right. Welcome to ChemTalks, online conference where scientists present advancements of their researches and give talks on the cutting-edge scientific themes. Today, our speaker is Professor Edward D. Antonio from the University of South Carolina, Beaufort. Let me briefly introduce our speaker. Dr. Edward D. Antonio is Associate Professor of Biochemistry and Structural Biology at the University of South Carolina, Beaufort. He holds a PhD in chemistry from North Carolina State University, where he specialized in biochemistry and analytical chemistry. His postdoctoral research at the University of Pennsylvania focused on structural biology, biochemistry, and bioinorganic chemistry. Dr. D'Antonio has extensive teaching experience, having taught courses in biochemistry, chemistry, forensic techniques, and data analysis, and many other disciplines also. He works remotely as an adjunct professor of biology at Jinan University in China and Feng Chia University in Taiwan. Dr. D'Antonio has received several awards and honors for his research, including the Outstanding Undergraduate Research Mentor Award and the American Institute of Chemists Foundation Award. Finally, he has 5 patents and has given numerous oral presentations on different conferences and symposia. So, the QA session will be held right after the presentation, and during the talk, you are free to leave your questions in the chat. Also, if anybody wants to give a presentation or talk on our channel, please write about it in the chat too. Professor D'Antonio, thank you very much for coming, and now the virtual scene is yours.
Thank you, Igor and George, for hosting me. I wanted to say it's always a pleasure to be on a chemistry platform such as ChemU and for ChemTalks. So let's get started with my presentation. The title of my talk is called Early Stage Drug Discovery for Chagas' Disease and Efforts Toward Drug Target Validation of Trypanosoma cruzi glucokinase. Chagas' heart disease is the cause of the chronic stage of infection. And what you can see from this figure to the top left is an image, is an X-ray image of a person who has Chagas' disease, but is at the very early stage where the heart is at the correct size that a normal person's heart would be. But as you progress to panel B, top right, and then going to bottom left, panel C, and then bottom right, panel D, there is a progressive increase in the size of the heart for folks who get Chagas' disease, and it is what is killing them because they're going to have arrhythmias and so forth. And this is one of the main chronic disease stages. Other stages, other disease manifestations could be that there are enlargements of other organs in the body, like the esophagus or the colon. And in those cases, it would just be that they would just get much larger from the irritation of the parasites. So what is Chagas' disease particularly? It comes from a protozoan parasite known as Trypanosoma cruzi, and that's what's shown here in the upper left figure. These are Trypanosoma cruzi trypamastogotes. That's a life stage. And this is shown under a blood smear under a microscope. They are single cellular organisms. They are the etiological agent responsible for the disease. And in the world, 6 to 7 million people or cases are found in mainly 21 Latin American countries, but we are starting to see a spread of this disease in the United States, even Australia and Europe as well, and that is due to travelers. And I'm going to talk about that in an upcoming slide. In America, there was about 300,000 cases estimated, and that's an alarming figure, to be honest, because this is kind of like a silent killer type of disease. Folks don't know that they have it, and until they get to the chronic stage when they're exhibiting the enlargement of the heart is when they would know. T. cruzi parasites are primarily spread by the feces of a triatomine insect vector, as you could see here on the bottom left here of the slide. This is a species of insect called a Triatoma sanguisuga. It's a kissing bug, and the way they take blood meals is that they would bite a person and they quickly rub their feces on the bite wound, and the Trypanosoma cruzi, which is inside of them, inside of their gut, is transmitting into the bloodstream of the person that they bit. And the disease can easily be acquired in Latin America, so anywhere from Argentina all the way up to Mexico. Here's a geographical distribution of reported cases, and you can see, like in Latin American countries, you have most of the areas are highlighted in red circles, and we have some in Europe, Australia, US, and Canada for a very small amount. These kissing bugs, I want to just make a quick mention about them. They're found in tropical climates, so with global warming and so forth, the thought is that they are moving northbound, and that's why they could be getting into the southern United States, everywhere from California, that west, going towards central like Texas, and then all the way east to Florida. So there have been spotted cases, and even in the state that I live in of South Carolina, there have been these insects found that carry T. cruzi. But because we don't have many individuals who are infected, it's unlikely that it's spreading around so much here as it would be in the Latin American countries. Now let's suppose a person gets Chagas's disease and they want to go for treatment. They can go see a physician, and there have been 2 traditional anti-ChGlcK therapeutics available since the early 1970s, known as benznidazole and nifurtimox. Benznidazole is going to be your first-line treatment option because it's better tolerated. And any of these therapeutics are very effective against the acute stage. So if you were to get Chagas's disease very early, like you had an early onset of some symptoms that you've noticed, or you went to the clinic and found out that you had it, if you were on a course and you took this medicine, you could literally cure it very quickly. So then these medicines can work. But the problem is, when the disease manifests into the chronic stage, these medicines aren't going to work that well. They were developed over 45 years ago. They're prodrugs. So essentially what happens is they make their way into the body and they're inert. But when they get to the T. cruzi cell, they get activated because they're prodrugs. But then they cause cell death, and they could cause cell death to human cells as well, owing also to the intolerable side effects, which would include allergic dermopathy, anorexia, peripheral polyneuropathy, vomiting. They're not fun drugs to take. They are going to cause a lot of bad side effects. And as a result, you get poor patient compliance from that. So there exists a need for new and improved therapeutic drugs. The DNDi, it's an organization that helps fund and for the research of new pipelines of drugs. That they have new drugs that are in the pipelines, but still, what a physician can use, like just as of today, would only be these 2 molecules or 2 compounds. So there really needs to be some work through to make developments. And there's also the fact that benznidazole, there is resistance concerns because T. cruzi have now been spotted to have resistance to such therapeutics. And that's where I got involved in the research. And I've been doing it for the past 10 years. Now, there is an enzyme found in the T. cruzi cell called Trypanosoma cruzi glucokinase. And I'll be calling it TcGlcK. And here's a quaternary structure of TcGlcK. It was an X-ray crystal structure found in 2007 by one of my collaborators, Dr. Artur Cordero. And this has a dimer. And I want to just talk about TcGlcK and then make a little reference to Trypanosoma brucei hexokinase 1, that's TbHex1, because, well, let me get started. I'm just going to read off the slide right here. In the cell biochemistry in Trypanosoma cruzi, you have a TcGlcK, and then you also have a TcHexokinase. So you have two glucose kinases that are present, and they are the first enzymes found in glycolysis. The X-ray crystal structure of TcHexokinase has still not been solved, and the metabolite called G6P, or glucose-6-phosphate, it has an important role in three metabolic pathways, such as glycolysis, the pentose phosphate pathway, and gluconeogenesis. In Trypanosoma brucei, which is, you could think of it as like a different protozoan parasite that causes human African trypanosomiasis, or we call it sleeping sickness, which is found in the sub-Saharan African countries. In that protozoan, I wanted to just make a little bit of an aside. That has TbHexokinases 1 and 2, these two isoenzymes, but it does not have a TC, or like a TcGlcK equivalent. And those are the first enzymes of the glycolysis in that protozoan parasite. And furthermore, TbHexokinase 1 is a validated drug target because it was confirmed by RNA interference. And I wanted to make a mention that TcGlcK could not be confirmed as a drug target by RNA interference because the machinery for RNAi could not work for this parasite. And when I started the research back in 2013, I chose this because it had a lot of key characteristics as the TbHexokinases that were validated drug targets. And it made sense to study it, but it was only referred to as a potential drug target. But nevertheless, I want to make that mention up front because it's going to lead into a much larger story as I go through the slides. Right. But just taking a look at structure of TcGlcK, here is the active site from the X-ray crystal structure, and the way that it carries out its enzymatic mechanism is that glucose is going to bind in the glucose binding site, and then ATP binds in its binding site. And the 6-carbon hydroxyl performs a nucleophilic attack with the gamma phosphate of ATP to then steal that phosphate and get converted into glucose-6-phosphate. That's the product. And you also make ADP. And in a T. cruzi cell, this is showing you 3 different pathways, such as glycolysis, gluconeogenesis, and the pentose phosphate pathway. But the important part is that you have that metabolite known as G6P. And that is the key because it's interconnected between these 3 metabolic pathways. And if we could stop TcGlcK from producing that G6P, we can constrict the flow through glycolysis and cause cell death. So we were looking to create inhibitors for TcGlcK to then create anti-Chagastic drugs. And this is an early stage drug discovery approach. Now, in order to do that, I first started out with a structural evaluation of TcGlcK, and I made a comparison with Homo sapiens hexokinase 4, or we can call that human glucokinase. In a program called ChemU, it's an X-ray structural program. I've done a secondary structure mapping or matching superpose, where I could superimpose TcGlcK over HSHXK4, and they have an 18% protein sequence identity with respect to each other. But I wanted to take a look inside of the active site. I had this idea one day where if I could come up with some kind of inhibitor that could dock inside of TcGlcK, because I've noticed that its active site was much larger than the active site of human glucokinase. And you can see here, the blue ribbons represent the human glucokinase, but the brown ribbons represent the TcGlcK. And the yellow carbons of this molecule here in the middle of of the active site right there represents this compound to the right called H-pop glucosamine, which is a potential inhibitor. And I hand modeled H-pop glucosamine into the active site of TcGlcK by essentially superimposing the glucose moiety onto where glucose was found in an existing crystal structure. And there is a linker with a amide linkage that goes into a phenyl ring, would extend into an outer part of the active site. And what I noticed when I looked very closely was that if you look up here through the molecular modeling at the human glucokinase, the H-pop glucosamine would essentially bang into a lot of amino acid side chains like lysine 56, glutamine 286, a threonine 168, asparagine 166, and so forth. So there was a lot of steric clashes observed if this molecule were to bind in human glucokinase. But if you were to hand model it into TcGlcK here in the bottom figure, which is color coded brown, you don't see many steric clashes. And with that, I thought about that we could go and proceed and get a hold of this molecule and see if it would bind more selectively to the Trypanosoma cruzi glucokinase homolog, because that's a goal in parasitic drug discovery, is that if you have a homolog that's found in the human cells, and in the human cells, we actually have 4 isoenzymes. We have human hexokinases 1 through 3, and then we have human glucokinase, which is called human hexokinase 4 for reasons that we can go beyond this presentation. But nevertheless, let's just say that human has 4 homologs. We want to create an inhibitor that would bind more selectively to the parasite compared to those 4. And furthermore, human glucokinase is a good representative example of those other 4. And that's why I chose it to do a structural evaluation. So circling back, I just wanted to tell you one more time that with all the steric clashes that we see in this theoretical binding scenario, in reality, you wouldn't have H-pop glucosamine bind in human glucokinase because there are these steric clashes. It would be prohibitive. But in TcGlcK, it should. And that was the running hypothesis, and then I wanted to test it out in the lab to see if that was the case. Now, the assay to study something like this would involve D-glucose plus ATP in the presence of your glucokinase would create glucose 6-phosphate here in chemical equation 1. The glucose 6-phosphate would then go into a second enzyme called glucose 6-phosphate dehydrogenase, also known as G6-PDH, plus NADP plus to form NADPH and a gluconolactone product. The NADPH is a chromophore in UV visible spectrophotometry, but it's also a fluorophore in fluorescence, having an absorbance maximum of 340 nanometers or a lambda maximum. But in fluorescence, it would have a excitation at 340 nanometers, followed by an emission at 485 nanometers, and it's very sensitive. It's an excellent kinase assay to do. So we went with that one to study this. Now, in addition to H-pop glucosamine, which is this third molecule you see in this figure, well, before you synthesize a molecule, because it was not available by any vendor, if you could synthesize, if you could purchase a molecule, you would want to do that first. So I found some analogs that were commercially available, and those were the two molecules shown here on the left. One is called benz-glucosamine, and the other one is called CBZ-glucosamine. And CBZ-glucosamine looks very similar to H-pop glucosamine. And when I saw that, I was very excited because we immediately purchased them and tested them in the lab. And we had some good results, which prompted us to then synthesize H-pop glucosamine, which was at the original starting compound I had. And then what came later was the synthesis of a more bulkier side chain called DBT glucosamine. So the last two here, H-pop and DBT glucosamine, we had to synthesize those in our lab, and then purify that by HPLC. With CBZ glucosamine, when we ran the assay and we looked at a Lineweaver-Burke plot, we found that it had a submicromolar inhibitory constant. So the Ki was equal to 0.71 micromolar. And the golden rule is if you have a Ki value in the nanomolar range, so this would be about 710 nanomolar, pharmaceutical companies would take interest in new drug molecules as such. And this one got us very excited as a competitive inhibitor because the Lineweaver-Burke plot shows that mode of inhibition. And we also were able to get the X-ray crystal structure of a TcGlcK CBZ glucosamine complex in our lab at a high resolution of 2.5 angstroms. And here's a simulated annealing omit map of CBZ glucosamine bound in the active site, where you have a lot of glucose binding residues interacting with the glucose moiety. And then over here where the linker is, you have an amino acid side chain called phenylalanine 337 that appears to be making some pi-to-pi stackings with the benzene ring of CBZ glucosamine. And these kinds of interactions are important to think about as we went to move forward, because I wanted to understand why this molecule was very good at binding in TcGlcK, and to understand like the entire active site and to look at all of the different amino acid side chains. It's a fun thought experiment, but it's also good to know that if you're trying to develop new molecules that would inhibit the protein even better. So back in 2015, when we published this work, I made a prediction that the reason why CBZ glucosamine would bind so strong was because you're also getting a pi-to-pi stacking between the benzene rings of phenylalanine 337 and that of CBZ glucosamine. But recently, we did a whole mutagenesis approach where we would take a look at F337 and turn it into smaller amino acids, okay, and actually disrupt that phenylalanine to turn it into something different, like a leucine. So we made a F337L mutation, which would hold the hydrophobic intermolecular interactions, but it would disrupt the ring system. And if it was due to the pi-to-pi stacking, kind of like reminiscent to what you would see for base stacking in duplex DNA, where nitrogenous bases would be at a distance of 3.4 angstroms. I'm just going to go back a slide. The distance from the ring of F337 to the ring of CbZ-glucosamine is a distance of 3.4 angstroms, and that's a key pi-to-pi stacking distance. And you see that same distance in duplex DNA. So I really thought it was because of that interaction. But when I did this whole mutagenesis study where I progressively diminished that F337 to different amino acids like leucine and then finally alanine, and I looked at the inhibitory constants with CbZ, the F337L was very similar in Ki to the wild type, suggesting that pi-to-pi stackings were not really at play. That was not the major reason for why it was a strong inhibitor. In fact, it seems to be because of hydrophobic forces. And finally, with the F337A, turning it into more of a whole, the Ki went up, so it lost a lot of, it had a weaker binding in there, 4.2 times weaker. Now, returning back to benz-glucosamine, CbZ-glucosamine, HPOP, and DBT, I wanted to take a look at how those inhibitory constants fared with respect to TcGlcK versus human glucokinase. And we had some very interesting results biochemically. We were able to get the X-ray crystal structures in TcGlcK for all 4 of these compounds. But more importantly, HPOP glucosamine, which was the original compound that I studied, that I did the molecular modeling at my computer, it had a Ki value with TcGlcK of 1.3 micromolar, but in human glucokinase, it had a Ki value of 242 micromolar, giving rise to a selectivity ratio for TcGlcK at a value of 186, which is very high. And for CbZ, the selectivity ratio was 245. And this other compound here called benz-glucosamine had a selectivity value of 12. After that, we went ahead and did some cruzi in vitro biological studies where I have a collaborator at New York University, and her name is Dr. Ana Rodriguez. And what she does is she has cruzi parasites that she works with in her lab. And she has created an assay where it's a transgenic luminescence assay. So essentially, she's using a firefly luciferase gene that's incorporated in her cruzi, and it's going to give off a luminescence signal for, a relative luminescence signal for a parasite found, so that they don't have to count the parasites under a microscope. And with that assay, I would send off my benz-glucosamine. Well, I would send off all of these 4 compounds, but I found out that the compound that had biological activity was not CbZ-glucosamine, it was not HPOP glucosamine or DBT, it was benz-glucosamine. and it had an IC50 value of 16 micromolar. And anything under an IC50 of 50 micromolar would be considered biologically active against this parasite. And to put it into perspective, the IC50 value for one of the clinically available drugs, benznidazole, if you look to the top right of this graph, the IC50 is 1 micromolar. So we landed on one that is 16 micromolar. And that was very interesting. It's modestly potent. And after that study was completed, I formed a collaboration or a partnership with Dr. Artur Cordero back in 2016. And we wanted to do a high throughput screening campaign of TcGlcK based off of these promising results. And the assay that we used in this high throughput screen campaign is the one that you see here. It's essentially the same assay, except that we're adding a resazurin step 3, that turns into resorphin. When NADPH reacts with resazurin, it turns into resorphin, which is a very bright emitting fluorophore. And Dr. Artur Cordero, he has a high throughput screening facility in Campinas, Brazil, at Centro Nacional de Pesquisa em Energia e Materiais, Laboratório Nacional de Biociências, also known as LNBio. So national lab over there, they have a lot of different facilities like X-ray crystallography and so forth, cryo-EM, things like that. Now, in the summer of 2016, we looked at a screening library. Well, there were 2 screening libraries. We have 10,000 molecules from a diverse synthetic library from a company called ChemTalks. And ChemTalks also has a natural derived library. So there's a lot of natural products. So it's like a compound library of 3,040 compounds. We pooled them together to give us all these compounds, and we did a primary high throughput screen where the compound concentration was 20 micromolar. By doing that, we landed on 56 unconfirmed hits. And we wanted to make sure that these unconfirmed hits were not interfering with the glucose-6 phosphate dehydrogenase and the diaphorase enzyme-coupled reactions that are in this high-throughput screening campaign. So that's called the confirmatory assay. And after doing that, we learned that we have 25 on-target confirmed hits. So that actually filtered out some compounds that may have been interfering with our detection scheme. I then resupplied these 25 on-target confirmed hits, and we ran further studies on them to see how well they work on target inhibition and some trypanocytal activity against T. cruzi, and then we also looked at host cell toxicity. We wanted to see if, because when we do the, when my collaborator, Ana Rodriguez, when she does the T. cruzi in vitro studies, she pre-infects NIH3T3 mouse cells. So that's our host cell. So that those mouse cells are infected with T. cruzi, and we wanted to see, of the winning compounds that came out of this HTS campaign, did they actually damage and cause cell death to those mouse cells? In the end, we landed on 2 hit to lead candidates. All right, these are the 25 compounds of interest that resulted from this screening campaign. We have singletons, but we also have core clusters that resulted, and they're they're compounds that don't look like anything that I did when I was doing the competitive inhibitor structural evaluation that I previously just showed. So that was interesting for me. It was that we could, there's more to this than just the competitive inhibitor that I thought about of H-pop glucosamine. But these two hit to lead compounds was exciting even more, because the cruzi EC50 from them, and these are called 3-nitro-2-phenyl-2H-chromine compounds. The EC50s of cruzi are as low as 2.1 micromolar for the one on the left, and 2.9 micromolar for the one on the right. They aren't really affecting the NIH3T3 mouse cells. And going back to compare to benznidazole has an IC50 of 1 micromolar. So very similar to the anti-Chagas properties of benznidazole, and it's a different molecule. More importantly, the TcGlcK inhibition is very much aligned to the cruzi EC50 value. So that suggests that you're not getting off-target effects. It's going only for this enzyme. So with that led us to another small library, 13 compound library focused screen, where I went ahead and found 3-nitro-2-phenyl-2H-chromine analogs, mainly through PubChem. There's a library called Zinc. There are these like online libraries that you can search by just constructing them, or just kind of putting the molecule. You can use Smiles codes. Those are usually very helpful to do. And you run it through the search, and then it looks for libraries that are sold by vendors, or compounds sold by vendors. And I was able to find 11 other compounds from these two compounds that I found. So the 11 plus 2 would make this 13 compound library, because I wanted to use those two as controls. And when I looked at cruzi glucokinase, we're also getting some really good inhibition. So it was not like a fake result. And, because we're seeing the same kind of activity again. So it's reproducible. And I like that. I love to see reproducibility in the work. And another interesting aspect to all of this was that Dr. Ana Rodriguez, she not only looks at cruzi parasites, but she looks at other diseases too. Like she looks at the parasites that cause sleeping sickness, malaria and leishmaniasis. Those are some other neglected tropical diseases. But I had her run Trypanosoma brucei, and I found out that all 13 of these compounds present some really good anti-trypanosomal activity against Trypanosoma brucei. So it acts as like a surrogate. Like the TcGlcK is acting like a surrogate to the T. brucei, which is outstanding. And we're ready to publish this paper. These are the 13 compounds that we looked at. Now, compound 1 and compound 9 here on this chart show the winning compounds of the high throughput screen in Brazil, but the other ones were the ones we purchased. So again, these are the 3-nitro-2-phenyl-2H-chromine analogs from their structural view. And this shows in this chart the kinds of IC50 values that you would get for T. cruzi and T. brucei, shown here in the last two columns. And particularly for T. brucei, I mean, we're getting IC50 values in the nanomolar range for a lot of these compounds. They're not as low, because the lower the IC50 value, then the better anti-trypanosomal activity it would have. They're a little higher in value, but they're definitely on the lines of benznidazole for T. cruzi. So this is some exciting drug discovery work. But I want to return back to what I was talking about in the beginning when I was mentioning cruzi as a potential drug target, where cruzi can't, it doesn't have the machinery to undergo RNA interference, so you can't really do genetic validation essentiality testing on it. You can with T-Brucei hexokinase 1, and they did that, and they were able to find that out, some scientists. But so the question that remains right now is TcGlcK a potential or is it a validated drug target? I mean, clearly I'm finding molecules that suggest that it is a validated drug target. So let me walk you through some parts of this dilemma. Because in the pharmaceutical industry, the pharmaceutical companies do not want to work with an enzyme or drug target that is not considered a validated drug target, which means it needs to go under a high scrutiny of investigation to say, if I put a drug into a person, or a therapeutic drug into a person, I need to make sure it's binding to that enzyme and not any other enzyme, because then that is like a liability, and we don't want that. Well, TcGlcK may be considered a chemically validated drug target based on the fact that there are now various studies, principally from my lab, that illuminates on its inhibition with parallel anti-T-cruzi activity. However, without genetic essentiality testing, it is not conservative to say or to classify the enzyme as a validated drug target, because genetic testing is like the gold standard. So it unfortunately remains as a potential drug target. Okay, let's add more to this story. All right, back in, I want to say 2017, there was a research article. Well, there were like 2 articles that came out from the laboratory of Dr. Roberto Docampo at the University of Georgia, where he used CRISPR-Cas9 to knock out a motor protein of the flagellum of the Trypanosoma cruzi. And if you don't knock out the motor protein and you look under a microscope, the assay is that the movement of the parasites are relatively fast. If you're just observing, and you can look at a movie of that. They have a movie image in their supplementary information in one of the publications. But on the knockout, where they knock out using CRISPR-Cas9 one of those motor proteins, looking at how fast they move is substantially slower. And that was like a key paper that basically suggests that you can use now CRISPR-Cas9 to see if you have a validated drug target or not, and that would be a genetically used tool here in the trade. Now, I didn't want to go down that hole because I'm not a molecular biologist, and it would just be like have to learn too many more different things, and I'd rather stay in my field of biochemistry. So I had some really interesting discussions with one of my collaborators. His name is Dr. Gustavo Mercaldi, but I also had discussions with Dr. Docampo at a conference. And both of them recommended to me that I could do something called a bump mutagenesis approach. And this is a biochemical and molecular biology approach. And it has also been conceived of in order to allow for validated drug target determination. So it is critical to move into the direction of solving this problem. And what I'm going to show now would be looking at, we're going to solve this problem. We're looking to know, is TcGlcK a validated drug target? And we're going to do it by bump mutagenesis. And that's what we're working on right now in my lab. So let me explain what bump mutagenesis is so you can get an idea. I was talking about earlier about whole mutagenesis, so imagine you have phenylalanine 337, which is very bulky, and then you progressively diminish the size of the amino acid side chain going to like a leucine, let's say a valine, and then an alanine. You can even go smaller to a glycine. So that's just like a progressive diminishing. If you have different mutants like that, that would be like a whole mutagenesis because you're creating a hole there in that active site. But a bump mutagenesis would be the opposite. It would be you have an amino acid that fits a certain size, but you could expand to much bulkier side chains. Like something is the biggest side chain would be tryptophan, and you could create a mutant that has tryptophan for one of these amino acids, and what you could do is you could take an inhibitor that's a competitive inhibitor like CbZ-glucosamine, for example, that fits into the active site cavity. But if a tryptophan is now present, which it originally wasn't, it would not allow it to bind because of steric hindrance. And that would be known, like if you could get that to work, that would be called a bump. Like you would bump it out. So here's the idea of how you could use that kind of concept in terms of creating, for drug target validation studies. Right? So the goals of this would be that we would want to take TcGlcK, like of an enzyme bump mutant that does not allow the inhibitor to bind well at all. And then we would, if we could get to that level, we could then genetically engineer T. cruzi parasites to include this TcGlcK bump mutation in its genome. We would then run an in vitro biological assay in the presence of inhibitor, like let's say we're using CbZ-glucosamine with the mutated T. cruzi cells. And then we would run an in vitro biological assay in the presence of inhibitor with standard wild type T. cruzi cells. Now, expectation number one would be that the inhibitor and mutated T. cruzi cells should not result in any cell death. That would be our test. And the second expectation would be that the inhibitor with the wild type T. cruzi cells should indeed result in cell death, and that would be our control. But this competitive inhibitor would need to be biologically active, and I was just saying CbZ-glucosamine as an example. However, CbZ-glucosamine was not biologically active of the 4 compounds that I looked into earlier. But we do have one, it's benznidazole, so that would be a great inhibitor to look at if we can test that. Determine a TcGlcK mutation to sterically hinder a competitive inhibitor from the binding in the active site. So there are some important Michaelis-Menten parameters that would be under consideration if you do a study like this from the biochemical point of view. You want to make sure that your ATP and D-glucose binding affinities do not change. If they do change, that could affect how this mutated enzyme would be expressed in the T. cruzi cells. For example, you want to maintain the Km values for ATP and D-glucose. And you also want to maintain the rate of reaction in the bump mutation. So you want to have comparable Kcat over Km efficiency parameters with respect to wild type. And finally, the competitive inhibitor that you're selecting should be precluded from the active site cavity from the sterically hindered mutational side chain. So you would expect either a much higher Ki value than what you would normally get, or you should just observe no inhibition whatsoever. Now, what we could do moving forward, and this is the project that we're doing right now, there's option 1, and there's another option. I'm going to show it to you. In option 1, we're considering benznidazole as the competitive inhibitor of choice for this bump mutagenesis experimentation. Some quick facts. It has a TcGlcK Ki value of 32 micromolar. So we need to know that. And if we make the bump mutation, we want to see that Ki value of 32 micromolar can dramatically increase, so that we can say, hey, benzglucosamine has been bumped out. And that's basically through an enzyme inhibitor assay. Obviously, we could do this in other routes, like if we wanted to do isothermal titration calorimetry experiments to look at, KDs. We could do that as well. Just another set of data that could corroborate with all this. And it is biologically active, this benzglucosamine, having an IC50 in T. cruzi parasites, again at 16 micromolar. Option 2 could be that we could look at CBZ glucosamine, because CBZ glucosamine is a much larger molecule, and it would be easier to bump out something like this, just because you're going to run into some problems when you do an experiment like this, because you're trying to put a large amino acid side chain in a place it probably doesn't want to go into. So if you have a larger inhibitor trying to dock in the active site, you might be able to find a mutant that would, fit the bill. So again, CBZ is a large molecule compared to benzglucosamine, so it would be easier to prevent access in a bump mutation. However, if we go down option 2, if we go down this road, we would need to explore novel CBZ glucosamine derivatives to understand if they can be designed in a way to become biologically active against T. cruzi, because it's currently not biologically active. And I do have an understanding as to why it's not. And if you take a look structurally at CBZ glucosamine, it has a midsection that is a carbamate. And carbamates are not that stable molecules, because there are enzymes called esterases that can clip amide linkages. They can clip carbamate linkages. They're known to do that, and that might be what's going on when we do the T. cruzi in vitro studies. Is that there's probably a T. cruzi esterase that's clipping it. But if you look at those 4 molecules I had, the DBT glucosamine also carries a carbamate midsection. Benzglucosamine, a little bit different. It has what's known as a benzamide midsection, and HPOP glucosamine has an amide midsection. So the benzamide, is probably a very stable. I'm sure, like an esterase is having trouble degrading that, and to try to create glucosamine, for example, as the free sugar. So what we could do here is that we could take a look at alternative motifs as amide or carbamate isosteres. And you could do what's known as an isosteric substitution at the midsections of these glucosamine analogs. So the purpose would be to create an amide or carbamate surrogate, where we would increase resistance to hydrolyzable peptide bonds of these presumable esterases. So this is all like in theory. I don't know if the esterases are really at play here, but it's just a hunch, but most likely what's going on. And finally, we want to verify that the carbon-2 linkage from the glucosamine, it properly extends into the outer part of the active site of TcGlcK, making that contact with phenylalanine 337. So we have to maintain geometry that is found within the geometric architecture of the ligand. Here's CBZ glucosamine shown here to the left, and these are 3 proposed derivatives of CBZ that we could make that should be hydrolysis resistant isosteres. This is a a sulfonamide here, and then you have a trifluoro. Basically, it's a carbon group with 3 fluorines bound, CF3 group, and there's another, like a CF2 group on this 3rd compound, which, if you take a look at the carbon of the amide on CBZ-glucosamine, well, actually on the carbamate, it's a sp2 hybridized carbon giving rise to 120-degree bond angles. The idea of these isosteres is that even though the sulfur of sulfonamide, that it has 4 electron domains around it, it doesn't appear to be like a 120-degree bond angle. It's, being isosteric like that means that it corresponds very closely to the bond angle that you would have for CBZ-glucosamine. And same for the carbons found on these other derivatives that contain the fluorines. Normally, you would expect tetrahedral for the carbon that connects to the nitrogen atom, where those angles would be close to 109.5 degrees, but they're going to be more towards 120 degrees. From the Cambridge Structural Database, if you look at some molecules down down these lines, you're getting bond angles upwards of like 115 degrees and so forth. So that was the idea where, okay, it's like make a 120-degree bond angle or something close, but that allows your aromatic ring to extend properly, so it makes that hydrophobic contact with F337. Now, in concluding remarks, although many inhibitors have been discovered to inhibit TcGlcK and exhibit biological activity against T. cruzi, it is still a very urgent matter to determine whether the enzyme is a validated drug target so that there would not be off-target effects when testing possible therapeutics. We have determined a bump mutation for TcGlcK, and it causes CBZ-glucosamine to indeed preclude access in the active site. So we are onto a bump mutation that we use CBZ-glucosamine as a test competitive inhibitor molecule, and it indeed bumped it out. So we are very happy with these results. Now, to continue with this is option 2. CBZ-glucosamine derivatives will be tested for in vitro biological activity. We are currently having them synthesized by collaborator Dr. Joshua Pierce from NC State University Department of Chemistry. He's synthesizing these molecules for us, so we're very happy to have him on our team. Benznidazole will also be investigated with TcGlcK bump, with this particular TcGlcK bump mutation, because of the fact that it has been determined to be biologically active. So if we can get another example into the picture, that would be amazing. So this is option 1. And finally, we will need to form a collaboration with a researcher who can perform the genetic engineering of T. cruzi to install this particular bump mutation and run the final assay experiment. Again, I didn't want to go out of my bounds into more of the molecular biology of this and working with parasitic T. cruzi. I'd like to just stay in bounds and work on the biochemistry of things. So it made a fun study, and we hope that in the future that we can confirm that it is a validated drug target, and that would be a great story for all of this. So I want to thank you for watching and being in attendance to my presentation. And I'd like to acknowledge the current USCB undergraduate researchers that are in my lab that work on Chagas' disease drug discovery. Those include Lindsay Baker. She's an excellent student, works on the bump mutation, and she's always excited to be in the lab and run the assays. So I'm very happy to have her in the group. And her partner is for this project is Anna Husted, and I have a new student named Carson Fry. And I'd like to thank all the folks who have given me funding in the past, particularly the Office of Vice President for Undergraduate Research at University of South Carolina. Thank you.
So, thank you, Professor, for this nice presentation. Guys who are watching us, you're free to drop your questions once again in the chat. The question which was already answered is is the following one. Have you tried to calculate binding free energies for your hit to leads?
No, I haven't. My approach is not down that avenue yet. I do like this kind of approach though, because we are not a computational or let's say, but I like this biophysics type of strategy, and I would like to move forward into some stuff like this. Thanks for the question.
The next question will be from my side, as there are no more questions in the chat. Probably someone will ask them as you're replying. Since I'm also a computational chemist, and from my perspective, it could be interesting. So I just want to know your opinion. So you remember the slide with CBZ glucose imine, where you analyzed the different non-covalent interactions of the molecule with the surrounding protein, and you try to assess which effects have which contributions. What do you think, using symmetry adapted perturbation theory, could quantify those effects? Are you still interested in knowing exactly which effects play the major roles and to know the exact numbers for these effects?
Oh, sure. Yes. There is actually also some other X-ray crystallography techniques that can look into things like this, and to know what you're mentioning is can even be like we're about to publish something on this with those intermolecular interactions, but we, if you're interested, we could even do more to it. It doesn't have to end right there, because sometimes when you uncover something, you open up another layer, and you might find something more interesting than what you think you might have. So it's definitely room to explore.
Thank you for your reply, and I would like to have one more question regarding the early stages. So the same slide, exactly the same slide as for the previous question, where you listed 4 different molecules which had quite similar structures. How did you come up exactly with these structures? And did you do some virtual screening or something like that to pick them, or it was just pure intuition?
Yes. So I'm kind of old school when it comes to looking at like I use hand modeling basically. I just, in my mind, I have the chemistry, and it's like from everything that I've ever read, it it's basically very simple concept. Make the glucose, basically make the glucose moiety, it will bind in the glucose binding site. Okay, let's start with that. That should be your very first objective. Then make a linker, and I chose a linker that had methylene groups, CH2. Extend it until it would hit something that it could make some hydrophobic interaction. Ideally, I was looking for coulombic attraction. For example, if you had aspartate, could we put like a positive charge to make a, sulfur, or to make a salt bridge, for example? We have a program through PyMol, and it's called ABTS, I believe, and it can show like charge. Basically, it shows you just like positive charges, negative charge, and neutral characteristic of the landscape of the surfaces. So, I mean, I am using some things like that sometimes, but in this case, it was purely hydrophobic going into the outer part of the rim there. So I wanted to say, like an H-pop would be probably a good choice. I put the hydroxy on the para position of H-pop because I wanted to have more water solubility. And also our HPLC, when we purify this compound, can give us a detection through UV. Um, Sometimes the choice has to depend on what you can detect when you're purifying it. This type of compound is not that easy to just, do a synthesis and then do a recrystallization, for example, like doing some organic chemistry. It's not that simple. Like it really requires, because it has a glucose moiety, so you have to run it through a Hillick stationary phase column in HPLC, and then you have to detect it. So you have to use that phenyl ring. It's going to be what absorbs the best for the ultraviolet. But had we gone some other directions, I would probably need to reach out to some organic chemists. But going back more to your question though, like because I did kind of go off a little bit on that question. I use just general intuition, hand modeling, and I do that for a lot of my studies, and it tends to work out. Now I know it is considered a slower method compared to um, some computer aided drug discoveries, but it's one that I feel comfortable with.
I see. Thank you very much for the reply. As soon as there are no more questions in the chat, I will use this opportunity to ask the last question of mine. So the active site of this kinase, did you discover it, or it was already quite established thing? And did you try probably to find some cryptic sites which were not established previously?
In 2007 was the identification of the first X-ray crystal structure by Archer Cordero. Now, on that landmark paper, I came into the puzzle in 2013, so that was, just some years later. Okay, there hasn't they in Archer had determined the structure of the active site at that moment from his crystal structure, but there wasn't much work done at all on this particular enzyme. But when I got involved, then I went right into that superposition with the human glucokinase and started to understand and discover that there is more of that steric hindrance. So, can you please say some of the second part of your question? I
Um,
do.
did you consider any search for cryptic pockets?
Yes, cryptic. Yes. Um, Thank you. I heard about cryptic sites, which I believe you probably would need to use NMR as your mainstay for noticing things like this. Maybe there's other sites or yeah, and also when we did the high throughput screening campaign and we came up with those 25 molecules of interest, they probably are they're not, they weren't determined to be competitive inhibitors. So they're binding elsewhere, and probably, like if you have an uncompetitive inhibitor or a non-competitive inhibitor, they're going to bind elsewhere from the active site. So then if you want to call that cryptic, then that probably, but I haven't identified where they would bind. So that's an interesting aspect and a very cool biochemical question.
I hope this discussion could inspire some computer-aided drug designers or someone else to model those binding motifs and calculate the binding free energies, as came from the question before. So there is a lot of huge field to discover. I guess this is all from my side. I mean,
May I say
for the
something along those lines?
sure.
Well, TcGlcK will not disappoint them because it has a very large active site. So there are, you know, there are possibilities that you might find new discoveries.
Okay. Thank you very much, Professor, for this nice talk. I hope you enjoyed, and our viewers also. And see you soon. Thank you.
Thank you, Igor. Thank you, George.